COVID-19 pandemic. All fundamentals described. Emergence, epidemiology, genome structure, diagnostics, surveillance, therapeutics, and vaccines. Production of biotechnology kiosk. Novel coronavirus, SARS-CoV-2, and global pandemic, the start and the aftermath. The novel severe acute respiratory syndrome coronavirus, SARS-CoV-2, COVID-19, was first detected in December 2019 in the Hubei province of China. Since the outbreak of this highly infectious disease, the COVID-19 pandemic has brought devastating and life-altering effects across the globe by infecting and killing hundreds of thousands of people in over 180 countries so far. SARS-CoV-2 can bind the angiotensin-converting enzyme 2, ACE2, on human cells with much higher affinity than the virus that caused SARS in 2003. The virus was later named SARS-CoV-2 because its genome sequence showed similarity with SARS-CoV, the virus that caused SARS in 2003. Some early research data suggests that the cell entry of coronaviruses depends on the process of binding of the viral spike S proteins to cellular receptors along with S protein priming by host cell proteases. Current research has indicated the need to focus more on understanding the cellular factors that are used by SARS-CoV-2 for the entry that could provide vital insights into viral transmission and subsequently lead to reveal therapeutic targets. Epidemiology, genome structure, and key viral factors. BAT was suggested in earlier studies as the potential reservoir of SARS-CoV-2. Detailed virus genome sequencing of COVID-19 was conducted throughout the genome to BAT-CoV RATG13 that showed 96.2 overall genome sequence identity of SARS-CoV-2. The virus genome sequencing results and evolutionary analysis suggested that BAT-CoV and human SARS-CoV-2 might share the same ancestor. Thus, BAT was a suspect as natural host of virus origin, and it was thought that SARS-CoV-2 got transmitted from bats via unknown intermediate hosts to infect humans. Further, researchers estimated the basic reproduction number, R0, of SARS-CoV-2 to be around 2.2, or even more, range from 1.4 to 6.5, in familial clusters of pneumonia outbreaks. This has led to a steady spread of COVID-19 across the globe by human-to-human transmissions, taking hundreds of thousands of lives so far. Researchers have divided coronaviruses, CoV, into 4 genera that include alpha, beta, gamma, and delta CoV. Among these different types of viruses, it has been shown that alpha and beta are able to infect mammals, while gamma and delta CoV tend to infect birds. The SARS-CoV-2 has been identified as a beta coronavirus, which is enveloped in a non-segmented positive sense RNA virus. In previous studies, researchers identified 6 CoVs as human-susceptible viruses. In these 6 CoVs, the group of alpha CoVs, HCoV229E and HCoV-NL63, and the group of beta CoVs, HCoV, HKU1, and HCoV-OC43, were shown with low pathogenicity, causing mild respiratory symptoms similar to a common cold. However, the 2 other known beta CoVs, SARS-CoV and MERS-CoV, have been shown to cause severe and potentially fatal respiratory tract infections. Some of the latest studies have clearly suggested involvement of the receptor angiotensin-converting enzyme 2, ACE2, that is used by SARS-CoV-2 to infect humans. Regarding genome structure and key viral factors, researchers have shown that 2/3 of viral RNA that is mainly located in the first ORF, ORF1AB, translates 2 polyproteins, PP1A and PP1B, and encodes 16 non-structural proteins, NSPs, with the remaining ORFs encode accessory and structural proteins. Further, the rest of the virus genome has been shown to encode 4 essential structural proteins, including spike, S, glycoprotein, small envelope, E protein, matrix, M protein, and nucleocapsid, N protein. It has also been shown that in addition to structural proteins, several accessory proteins can interfere with the host innate immune response. diagnostic and surveillance technologies to battle SARS-CoV-2 pandemic. During these difficult times caused by COVID-19 pandemic that has seen a continued spread of the disease, it is critically important that we have effective diagnostic tools and practices in place that can play a key role in the containment of COVID-19. Effective diagnostics can enable the rapid implementation of control measures that can limit the spread through infected patient identification and subsequent isolation. This also enables contact tracing, including identifying people that may have come in contact with an infected patient. Regarding the employed diagnostics of this disease, the computed tomography (CT) images that were obtained earlier during the time of disease breakout suggested pneumonia-like symptoms and abnormal lungs in infected people. The negative stain transmission electron microscopy (TEM) was used to identify the morphology of the virus, and the images revealed virus with diameter ranging from 60 to 140 nanometers that comprised an envelope with protein spikes and genetic material. Polymerase chain reaction PCR, especially real-time PCR, RT-PCR, has been employed as a highly sensitive and specific method for detection of infectious diseases such as COVID-19. The existing diagnostic practices are mainly based on real-time multiplex PCR, RT-PCR, which is considered the clinical standard for SARS-CoV-2 nucleic acid detection. RT-PCR is currently used for the diagnosis of COVID-19, along with screening with CT scans. A multiplex PCR panel of known pathogens was employed to analyze the samples from infected patients. This helped identification of the previously unknown pathogen as an RNA virus through next-generation sequencing. Researchers were able to use the whole genome sequence to develop PCR kits to diagnose patients suffering with COVID-19. To this end, PCR, RT-PCR-based methods have been employed to detect nucleic acid, example RNA, of novel coronavirus SARS-CoV-2 for diagnosis of COVID-19. However, PCR, RT-PCR-based methods have limitations due to the fact that they are typically restricted in a centralized clinical laboratory that requires sophisticated equipment and well-trained personnel. Further, the less availability of enough PCR reagent kits is another issue that has been faced to keep up with the high demand. In addition, another challenge is the reliance of RT-PCR on the presence of detectable SARS-CoV-2 in the sample collected. Because of this limitation, it is difficult to identify infection of SARS-CoV-2 in a recovered, asymptomatic patient who was infected with the disease before. So, this does not allow to enforce control measures. On the other hand, CT-based imaging procedures are quite expensive and require technical expertise that also cannot specifically diagnose COVID-19. It is of great concern worldwide that COVID-19 might persist well beyond 2020. This places a significant stress on the global healthcare systems. In view of this, it is believed that new innovations in research to develop next-generation diagnostics and surveillance technologies are needed to combat the disease. As for improving efforts on surveillance technologies, in addition to nucleic acid-based tests, serological tests using proteins are being considered. These tests are considered superior than nucleic acid tests taken alone due to the reason that they have the benefits of detection after recovery. This can be leveraged to help clinicians to track both sick and recovered patients, which can provide a better statistic of total SARS-CoV-2 infections. Thus, the ongoing efforts are focused on to develop new rapid diagnostics, including point-of-care diagnostics, and surveillance technologies to detect SARS-CoV-2 early in order to control the disease and mitigate it in a timely fashion. Diagnostic technologies for COVID-19. Looking beyond the current state of the art. More advanced diagnostics are believed to include serological tests, i.e., blood tests for specific antibodies-based technologies. These technologies are thought to be easier to administer that may complement nucleic acid tests for diagnosing COVID-19 infection. Researchers are paying a lot of attention to nucleic acid tests using isothermal amplification that are currently in development for SARS-CoV-2 detection. Isothermal amplification techniques include recombinase polymerase amplification, helicase-dependent amplification, and loop-mediated isothermal amplification, LAMP. They can be conducted at a single temperature without requiring specialized laboratory equipment that can provide high analytical sensitivities to PCR. RT-LAMP employs DNA polymerase with 4 to 6 primers to bind to 6 distinct regions on the target genome. Researchers have developed and clinically tested reverse transcription LAMP, RT-LAMP, tests for SARS-CoV-2. Current research has also focused on viral protein antigens and antibodies that can be used for diagnosing COVID-19. Among currently considered point-of-care approaches in diagnostics for COVID-19, lateral flow antigen-based point-of-care detection for SARS-CoV-2 is under development for diagnosing COVID-19. Another point-of-care approach based on microfluidic devices is being considered. Microfluidic devices are essentially based on palm-sized chip etched with micrometer-sized channels and reaction chambers. The function of a chip is to mix and separate liquid samples using electrokinetic, capillary, vacuum, and/or other forces. Microfluidics-based diagnostics for SARS-CoV-2 offer a number of advantages that include miniaturization, small sample volume, rapid detection times, and portability. All these point-of-care diagnostic technologies are envisioned to be adapted to detect SARS-CoV-2 RNA or proteins. Innovative mass surveillance and monitoring technology, preventing future pandemics. It is believed that a robust system of networks consisting of mass surveillance with rapid diagnostics can help public health officials monitor virus spread and proactively identify areas with increasing infections. Such a functional system can also be leveraged to anticipate surge capacity needs and deploy needed resources to the appropriate areas accordingly. However, the functioning of such a system will be impaired in absence of clear and transparent collaboration and communications between nations at the international level and between federal and state and principal public health laboratories, hospitals, government agencies, and communities at the domestic level. Researchers are considering to employ the innovative combination of diagnostics and smartphones that is believed to provide greater communication and surveillance. Smartphones are considered an attractive choice because they possess the connectivity, computational power, and hardware that can be facilitated for electronic reporting, epidemiological databasing, and point-of-care testing. It is believed that smartphones can be made a widely accessible technology to coordinate responses that include real-time geospatial information empowering national and global health agencies to implement coordinated control strategies during large outbreaks of COVID-19. Therapeutic innovations to control SARS-CoV-2 disease. Innovations in therapeutic strategies are thought to provide viable pathways to a faster solution to the current COVID-19 pandemic to cure infected people and also to prevent further epidemics. The promising strategies include therapeutic agents targeting nucleosides, nucleotides, viral nucleic acids, and enzymes and proteins involved in the replication and transcription of SARS-CoV-2. To this end, repurposing some of the existing antiviral agents to treat infection from SARS-CoV-2 are already moving into clinical trials. Researchers have shown potentials of approved nucleoside analogs, favipiravir and ribavirin, and experimental nucleoside analogs, remdesivir and galidesivir, against SARS-CoV-2. Previous studies have suggested that nucleoside analogs in the form of adenine or guanine derivatives can target the RNA-dependent RNA polymerase and block viral RNA synthesis in a broad spectrum of RNA viruses, including human coronaviruses. For example, researchers have shown favipiravir, T705, which is a guanine analog approved for influenza treatment, can effectively inhibit the RNA-dependent RNA polymerase of a range of RNA viruses including influenza, Ebola, yellow fever, chikungunya, norovirus, and enterovirus. In this regard, a recent research reported the promising activity of favipiravir against COVID-19. EC50 equals 61.88 micromolar in Vero E6 cells. Remdesivir, GS5734, which is essentially a 1-cyano-substituted adenosine nucleotide analog prodrug that inhibits viral RNA polymerases, has shown quite a lot of promise. This antiviral drug has been tested for a wide-ranging antiviral activity against several RNA viruses. In a most recent research, in vitro activity of remdesivir was demonstrated against SARS-CoV-2, and 2/3 of severe COVID-19 cases improved on administering remdesivir, In this study that employed a cohort of patients hospitalized for severe COVID-19 who were treated with compassionate use remdesivir, clinical improvement was observed in 36 of 53 patients, 68%, in the category of oxygen support, while improvement was observed in all 12 patients who were breathing ambient air or receiving low-flow supplemental oxygen. Further, 5 of 7 patients, 71%, who are receiving non-invasive oxygen support, NIPPV, or high-flow supplemental oxygen, also showed improvement. Only 8 of 53 patients, 15%, in the oxygen support category showed worsening health conditions. Innovations in the combination antiviral drugs seem to hold the key. To this end, a recent work showed the feasibility of the combination of remdesivir and chloroquine that was demonstrated to effectively inhibit the SARS-CoV-2 in vitro. Additionally, it was shown that treatment with chloroquine prevents the spread of SARS-CoV infection in the post-infection period. In this study, researchers employed clinical isolates of SARS-CoV-2 and evaluated the efficacy of 7 agents: ribavirin, penciclovir, nitazoxanide, nafamostat, chloroquine, remdesivir GS5734, and favipiravir T705 in vitro conditions. Cytotoxicity was also evaluated in Vero E6 cells, which was followed by infection of the cells with SARS-CoV-2 clinical isolates. The test drug was then evaluated at different doses. Reverse transcription PCR-based quantification was done to get the viral yield, which was later confirmed by immunofluorescence microscopy, nucleocapsid protein visualization. The results showed that both chloroquine and remdesivir inhibited virus infection at micromolar level, 0.77 to 1.13 micromolar, and with high selectivity. Convalescent plasma therapy in COVID-19 patients. Convalescent plasma therapy, a classic adaptive immunotherapy, has been applied to the prevention and treatment of many infectious diseases for more than 1 century. Patients who have recovered from COVID-19 with a high neutralizing antibody titer may be a valuable donor source of convalescent plasma. Nevertheless, the potential clinical benefit and risk of convalescent blood products in COVID-19 remains uncertain. Thus, researchers have explored the feasibility of convalescent plasma transfusion to rescue severe patients. Recent research results that were published from 10 severe adult cases showed that 1 dose, 200 milliliters, of convalescent plasma was well tolerated and could significantly increase or maintain the neutralizing antibodies at a high level, leading to disappearance of viremia in 7 days. Meanwhile, clinical symptoms and paraclinical criteria rapidly improved within 3 days. Radiological examination showed varying degrees of absorption of lung lesions within 7 days. These results indicate that convalescent plasma can serve as a promising rescue option for severe COVID-19, while the randomized trial is warranted. Vaccine developmental efforts. Development of an effective vaccine is the clearest path to controlling the COVID-19 pandemic. To accelerate vaccine development, some researchers are pursuing, and thousands of people have expressed interest in participating in, Controlled Human Infection Studies, CHIs, with SARS-CoV-2. Besides focusing on the entire virus particle-associated inactivated or attenuated viral vaccines, researchers are also employing the subunit candidates, such as S1 protein and/or the receptor binding domain, RBD element of SARS-CoV-2, that are considered as valuable targets for vaccine design. It is believed that a deeper understanding of the atomic level structure of the COVID-19 spike protein will allow for additional protein engineering efforts that could be leveraged to improve antigenicity and protein expression for vaccine development. This information could pave the way for precision vaccine design and ultimately discovery of antiviral therapeutics that would accelerate medical countermeasure, MCM, development. Moderna's vaccine candidate, mRNA-1273, had been cleared by the FDA to move into a Phase 2 trial. Moderna already announced today that their vaccine has induced persons' immune response. Moderna's vaccine candidate, which was developed in collaboration with scientists at the National Institute of Allergy and Infectious Disease, is made out of messenger RNA, hence the phrase mRNA in the vaccine's name. This molecule is responsible for carrying the genetic recipes for making different proteins to a cell's protein production factory. Regarding other vaccine candidates, volunteers in New York received the first doses of an mRNA-based vaccine similar to Moderna's, called BNT162. Produced by Pfizer and a German pharma company called BioNTech, BNT162 is one of 4 genetic vaccine candidates the two companies are jointly developing to fight SARS-CoV-2. The Phase 1 trial will enroll 360 people in 4 different research hospitals to see how safe these different variations are compared to a placebo. Other approaches for a faster vaccine development proposed by the World Health Organization for a speedier way forward with challenge trials.